from putting the picture on the slide to show you the complexity of the organization of the communities of strep salivarius. And of course, it's a model study. This is the cells that are attached to a HEP2 cell line, but it looks like all of them are happy. The eukaryotic cells are happy, and the bacterial cells are happy. This is the symbiotic type of relation. And since I mentioned several times strep salivarius, and again the K12, for instance, is the strain that was identified by Dr. John Tag and developed into the product. We know that it is producing numerous bacteriocins, salivariceins A and B, and it can actually target numerous pathogenic conditions in the oral cavity. Honeycombs, cystitis, oral thrush can produce anti-inflammatory effect, many functions. It has officially assigned GRAS status. Human trials were conducted. Genome was sequenced, and basically we do know that this is the formulation that helps maintain healthy profile and to remove pathogens from the environment. M18 is another microorganism from strep salivarius, produces 4 antimicrobial proteins. Human trials were conducted, and actually the good thing was that in these human trials the probiotic formulation assisted with the recovery from the pathogenic status, and in vitro it has been shown that the microorganism can inhibit strep mutans and pyogenes cells. Another thing is that if we look at the targeted areas, not only cases but also gingivitis, periodontal disease, anti-inflammatory activity, also the strep throat, and the strep throat big problem because you feel miserable, you are not quite sick to complain about it, but it's a persistent condition, and many individuals, especially younger age, but also in adults, these may go with people through their entire life. Now, another important thing, probably this is the right thing to mention here. I don't know if I will have a moment to recollect it in my memory later. Caries. Caries, to some degree, at least in the Western world, is declining. Periodontal care, periodontal health. This is something on the rise. Periodontal infection. In the U.S. alone, between 70 and 80% of population, and again, the statistical data, sometimes it's a very loose thing, but that's what's coming from some sources. Between 70 and 80% of population is diagnosed with different level of severity of periodontal health issues. This is a very big number, right? In some countries, caries is not that big of a problem. Why? Because fluoride-containing toothpaste that was introduced many years ago, and also in the formulations for children, which controls caries development. Other parts of the world that we do know examples where fluoride-containing toothpaste was not introduced a long time ago, 50, 60 years ago. We see still recurring cases of dental caries with relatively high numbers. Oral respiratory trials. I couldn't resist putting respiratory trials because these are the ecological niches which are connected. It's an open environment to some degree. Microorganisms can migrate from one area to another. And again, we have strep salivarius, otitis media, strep throat, 3 bacteriocins identified. And again, it's interesting how the science evolves. the original data which came from one reference didn't have the bacteriocins identified. And last year, 3 bacteriocins were identified in this formulation. All right. Safety, yes, these microorganisms are safe. In vitro, it has been shown the inhibition of strep pneumonia and pyogenes. And then again, recently it has been shown that both strains can inhibit biofilm formation and adhesion to the HEP2 cells. So definitely interference with the quorum sensing. And again, very new, fresh in vivo studies, 200 children were involved, and 64% reduction in the episode of upper respiratory tract infections compared to the control studies. So this is really a very good indication of health promoting activity for the Strep salivarius and the Rallis bacteriocin producing probiotics. So what do we have here? We have the situation in the oral cavity where different types of microorganisms are present, the bad guys, the good guys. They sometimes coexist, and in many cases they have to get into the battle. How we can assist this battle? What should be done? What can we do? And again, I repeat myself. Until now, I am firm and confident in saying that the approach where we talk about the use of complementary treatments, combinations of the conventional antimicrobials with probiotics, either given together, and in this case, probiotic bacteria must be resistant to the antimicrobials or medicinal formulations used, or sequential treatment. This is where the best results can be achieved. And with that in mind, I would like to finish our today's presentation, and I will be addressing oh, sorry, I moved. My goodness, I don't know how did that happen. Taking my words back. Why did it jump? All right, let me. Not quite. Good thing. Not quite. I was thinking, how could it be that I'm so early done and not seeing my beloved spore formers? Back on track. All right, beneficial effects, primary and secondary effects with the probiotic bacteria. 15 minutes to go. Give me my time back. I want it. All right, primary effects of probiotics, of course, colonization, production of antimicrobials, competition with the pathogens, reduction of their adhesion, modulation of the immune system, and production of the beneficial enzymes which contribute to the overall health. And the secondary effects, reduction of polyposis, reduction in the case of otitis media, disruption of the biofilm, even lower COVID-19 morbidity and infections, believe it or not. Candidate disruption, my guess would be yes, because we have examples from the clinical trials. Now, the situation with the challenges through the lifespan in the early ages, otitis media, then tonsillitis, then caries. In the mature adults, halitosis, gingivitis, sinusitis, periodontitis, and candidiasis can develop in the older population. Why? Because the immune system is declining. The production of saliva may be lower in the elderly population, lower saliva levels, less amount of histidines and defensins in the oral cavity, which are protecting, which are antimicrobial proteins that are protecting against oral candida, and candidiasis can develop. Finally, now my beloved bacilli. Periodontic bacteria, Parphyromonas gingivalis is the main one, and then Treponema, Tenerella, Privatella, and other nasty creatures to follow. So, before we go to my beloved bacilli, we need to understand what is gingival index. It was introduced in 1963. It can be used in all teeth or separate individual teeth, and a variety of surfaces, and also it can be identified and used as the parameter in erupted teeth, tooth retained roots, and many other lesions, many other areas. So scores and criteria from 0 to 3, from no inflammation to mild, moderate, and severe inflammations by observation. What is gingival index? How it is calculated? It can be calculated in individuals, excuse me, or it can be calculated in populations. Total score divided by the number of surfaces examined, or total score divided by the subjects, the number of subjects. And the scores are distributed between 0.1 and 1 mild, 1 to 2 moderate, and 2 to 3 severe gingivitis. So we need to understand these parameters because we will see them in the future slides. And the last thing we need to introduce is a BANA test. BANA is epithelial arginine naphthalate test, which is used to analyze some gingival microbiota. And again, we're dealing with the microorganism, periodontopathogen bacteria, Parphyromonas, Treponema, Bacteroides, which can also be involved in the production and the enzymes that can hydrolyze the substrate, which is benzoyl arginine naphthalenate butter. And BANA reaction can be used as an indicator of these bacteria presence, and it's kind of a qualitative method, not quite quantitative. The reaction is carried out with spurious canal bean cards, which are produced commercially, and naphthalate reacts with the dye, forming a blue-black color spots on a pale reddish-brown background. So, two probiotics, or two formulations, rather, I want to introduce. It's the probiotic microorganism and the probiotic-derived mouth rinse. Both came from the studies conducted in Japan. And the microorganisms used for these studies is Bacillus subtilis, DB9011. And why this microorganism is interesting, what is so special about it? This is a soil isolate. It has been shown to have beneficial effect in a variety of formulations for agricultural purposes. And investigators decided to look at the possible role of this microorganism in the oral cavity. And they wanted to compare the live cells against the fermented product. So the formulation was designed with our Rex tablets, which contain Bacillus subtilis DB9011. And the good thing about it, it's a small format. So the shelf life can be really very long. It's 300. The description is kind of murky because they're talking about the extraction of 300E. In some cases, they call it the cultured medium. In some cases, they say that they contain cells, 0.9 gram per 100 of Bacillus subtilis. But I think it was a typo in the paper. I had to mention that, right? So what we have here is some of the examples. The BANA score of the Vital Rex and dextrine. Dextrine, of course, is the control in these cases. Okay, and what we have is VL Vital Rex is really performing reasonably well compared to the control. All right, the E3, which is the mouth rinse, supposedly the fermentate of the Bacillus subtilis. And again, can see here, 14 days and 30 days E300 compared to the control, negative control here. Again, we see really very good performance. So take-home message, the probiotic and probiotic-derived formulations can perform really well, also comparing to the control. Leading profiles, and again, we have viral ranks compared to dextrine, day 14, day 30. The numbers are statistically different, which is again the good indication of the performance of the probiotic microorganism formulation. And these tablets there, the individuals can sole control of them, so they are dissolving in the oral cavity. Leading probe with the mouth reads, and again we have the comparison of the mouth reads against the control, and this is the green, which is the mouth read, so with the benzoyl chloride commonly used, and again we see the differences in the performance, not as impressive. So mouth read does work, but not as well. Gingival scores, and with the gingival scores, we again see the Viltorex versus the control, and the mouth reads, the fermentate of Bacillus subtilis against the control, and again we see here the positive results with the formulation of bacteriocins. Bacteriocins, the Bacillus. So, concluding remarks: factors that affect the development of probiotics for oral health. So what are the limiting factors? Lack of standardization, which makes it very difficult to compare and to replicate the results. And with the same studies conducted by John Tack, we recently saw an example, and the paper was published. They were trying to reproduce conditions, and they failed. And they said, oh, it's not working. And I think it was 2 or 3 years ago. And then the group of John Tack, the New Zealand scientists, thoroughly investigated the research, the experimental layout of another group, and they clearly indicated that the conditions were not observed as they should be, and that is why there was a failure. All right, a limited understanding of the mechanisms of action. In some cases, we see bacteriocins. In some cases, we see some kind of immunomodulatory activity. One of the slides you saw today mentioned the action of the cellular elements. So even if the cells are dying, perishing, the cellular components, and I brought your attention to the importance of postbiotic action, they can also contribute. So we have multiple variables which may deliver positive effect, and we don't know all of them. Inadequate clinical evidence in many cases, small size of the groups, and of course the challenges with the regulation systems. Probiotics are considered as dietary supplement rather than drugs. It's quite difficult to ensure the quality and safety of probiotic products. So this is something, the last bullet, something that is being taken care of as we speak. There is a progress in this area, but still more effort is required. This is finally my last slide. All right, thank you very much for your attention, and now the floor is open to the questions.
This was really a very informative, eye-opening session. Lot of good things, both the environments are like diverse in the vaginal case and the oral microbiota. And it depends, the kind of food habit and environment and the person who is living there. it itself creates a great diverse environment, and really replicating in this is challenging.
Right.
And we are like working on these probiotics and their metabolites. If using such probiotics can be correct this dysbiosis if it is happening through this, that will be a great deed. And a lot of things has to be done in this area. Yes, any question from the chat box? One I can see.
I don't see the chat box. Okay. Yes, I opened it. Right. Yes, I see. Is it true that baby born by C-section are at high risk of jaundice, while the normal ones are protected by vaginal microbiota? I will be careful saying that, especially saying high risk. And we do know that the C-section delivered babies, they restore their normal microbiota really very fast. Okay, we can say that within the week or couple of weeks, it's coming to restoration. And again, it depends on case by case. Is there higher risk? Yes, there is a higher risk. All right, but how much higher? I wouldn't be able to say with confidence, because it's a case by case. But yes, it has been proven that the C-section delivered babies have, not the same microbiota, much poorer than the babies delivered through the vaginal route.
So we don't have any other question in the chat box. If someone want to ask directly, otherwise we'll go for.
Yeah, I apologize. I don't know how it happened. It jumped, and I was like, "Oh, did I?"
Oh, you finished in 1 hour this time.
Yeah, I still finished on time, but I was like, I'm running early. How is that possible?
You had conclusion slide 2 times, somewhere in the middle.
There are some glitches, and the system shows glitches. I don't know how it happens, but anyway, I'm pushing the button and it jumps, back and forth, and I have to watch for what I'm pushing. Okay, so anyway, we have like maybe 15 minutes, right? And then we'll go to the next session for today.
Yeah.
All right. Good. So what do we have next? We have the pediatric health. In the meantime, I will change the slides. Okay, pediatric health. So we're still here, and if anyone has questions, of course, feel free to ask.
Yes, sir. Excuse me, sir.
Yes.
Sir, for treatment of any oral disease like periodontosis, which should be preferred, using probiotic therapy or using their bacteriocin for treatment?
Okay, bacteriocins were not shown as the formulations in clinical trials yet. But the probiotics that produce bacteriocins and these Strep salivarius strains, they did show positive effect. And what Dr. John Tagg is emphasizing is that these probiotics are producing multiple bacteriocins. So he is putting emphasis on production of bacteriocins. Now, why there is, and again, I have to be very careful saying that, for instance, bacteriocin producing probiotic can be better than bacteriocin itself. I must be very careful when making such, assumptions, claims. What I feel, and again, that's a feeling, is that when you deliver probiotic that is producing bacteriocins, it may have a good chance to establish its presence in the environment and to become a part of the resident microbiome. And if that happens, then the microorganism can have the production of the bacteriocin over the extended period of time, not only during the time of treatment. However, on the other hand, when you have the formulation of the bacteriocin, you may have the formulation delivered with a much higher concentration of the biological active substance, which may enable the action of this antimicrobial, in our case bacteriocin, at a much higher efficiency. Which way is better and which way is more effective at the end of the story, I don't know. We should not forget about other functions such as immunomodulation, suppression of the pro-inflammatory components of the immune system, and the competitive exclusion. Right? So, and again, don't get me wrong, bacteriocins can also work as quorum quenching molecules, right? So I don't think we have a capacity to do the qualitative and quantitative comparable analysis of two. But I mean, probiotics work. And it's a long story of John Tagg's personal involvement in development. And Bliss K12, and I am in no way affiliated with the company that is making and the marketing this probiotic. Bliss K12 is marketed all over the world. And this is the formulation with probiotics, Strep salivarius, that does help against the strep throat and other conditions.
So, Dr. Chicken, does this K12 and M18 are available in the market? Can we buy?
Well, I don't know about India. It's definitely on the market in New Zealand. You can buy it in the US through Amazon. So if there is Amazon in India, maybe Amazon. I know that it is produced also in Canada, I believe, or they distribute it through Canada. So yes, it is available. And whenever I talk to my students in the class and asking if any of them had strep throat or still have, usually few hands are being raised, and I'm then saying them, do try this formulation. I'm not saying that it will absolutely help, but there is a chance that it will help you.
So Pallavi is asking question because she's working on bacteriocins against oral health, oral pathogens. So we are looking at the possibly the probiotic bacteria were producing bacteriocins and how they can be applied in oral health. So
you know what I would do? I would probably take a piece of paper and make a chart left and right. What kind of contributions both of them can make. As I just mentioned, immunomodulation, bacteriocins are not really that much into immunomodulation. Quorum sensing inhibition, both of them can deliver because bacteriocins are produced by bacteria. So bacteriocin and the cell. And the cell, you have additional multiple factors. Not to forget that, for the bacteriocin to act, you have to have really well-designed delivery system. and I don't know what kind of systems you are considering. So whatever works in the test tube or a petri dish, it's a one story. But whether it will work, so you will need something, and I'm just thinking cloud. You will need a control release system. Okay, I would go with, for instance, I don't know, nanogels, nanofibers. I would go with something that you can use as lozenges. Something that will have the bimodal release kinetics. So first kick with a high concentration, then lower concentration to maintain the inhibition of the microorganisms in the environment, and we did these kind of studies, and the bimodal delivery of the antimicrobial is the most effective scenario. And
so overall, what do you suggest that the probiotics in oral environment and probiotics in gut environment, and see, they absorb which surface better? Like on the oral, you have a tooth surface, you have a muscular surface. So do they have capability to absorb or adhering ability to the tooth surfaces and perform their probiotic or antimicrobial activity?
Which one?
In the oral environment.
I wouldn't say better, because we're dealing with different microorganisms there. I'm pretty confident saying that in oral environment we definitely want Strep salivarius to work, and in gut environment we want lactobacilli. So it's like comparing apples and oranges. They're different types of microorganisms, and they do different job in different environments. And while lactobacilli are present in oral environment, they're not really that big numbers. So guess we can start. Hope that this time the slides will not jump.
Yes, you can go for next lecture.
Okay. Why did they jump?
Sorry, sir. Good evening, sir.
There is a question, Dr. Sikinder.
Okay, yeah, sure. Go ahead.
Just I interact with you, sir. Hello, respected sir.
Yes,
first of all,
you.
I thankful to you for a valuable and most important lecture given by you. I also thanks to my PhD supervisor, Professor Santosh Kumar Tiwari, for organizing this GIAN program. Sir, I have read your many papers and cited in our many research articles. I have learned many things from your articles, like how to design experiments and how to analyze our research data. Or I am also thankful to you for sending me your recently published review articles.
I can recognize your name. Thank you
yes,
for your nice
yes,
words.
Yes, sir. And I would consider myself lucky if I got a chance to work with you.
Okay. Thank you for your
Because
kind
I am
words.
also working on biofilm inhibition in the presence of bacteriocins like plantaricin and other bacteriocins isolated from different lactic acid bacteria.
All right. So what is your question, if you have any question? But it's nice.
Yes, sir. I want to interact with you.
Right. Okay. Yeah, we can talk later, and involving Dr. Tiwari, and these can be discussed and probably should be discussed. We will do that. And saying that I don't want to keep the rest of the audience waiting, we will go through the next topic today, which is probiotics in pediatric health.
Yes,
something wrong. Okay. Pediatric health, which is like a difficult, very challenging area, because these little creatures are rapidly evolving, developing microorganisms, microbiotas in every ecological niche of the developing body is changing. So this is really challenging, but very rewarding area if we can bring some health improvement to little creatures. That's priceless. All right, so the outline of our today's gathering on the pediatric probiotics, we'll talk briefly about microbiota. We'll then look what is happening with probiotics and whether they work from mother to child, and we can see some absolutely amazing, fantastic results, almost mysterious, with the long-term trials where the probiotic effect is extended for many years. Prophylaxis treatment for children, some hopes and disappointments, and the conclusions. So, so mother-to-child colonization, some of the take-home messages here. With regard to the normal colonization, we have kind of a germ-free intrauterine environment, which is not really true. Phase 1, acquired maternal vaginal colonic microorganisms. This is a normal delivery, then introduction of oral feeding, breast milk, and the microorganisms that are coming with the breast milk, winning, and then the last phase, the complete colonization. As I mentioned actually before, by the 1-year-old or 1.5-year-old, we have theoretically, if everything goes right, we have a complete colonization. All right. So the important thing, of course, is to look at the maternal factors, which may severely influence the intestinal microbiota of the infant during the first 1,000 days after the birth. Gestational age, of course, the full-term infants will have better developed microbiota, much quicker establishing into the normal healthy adult status as compared to the premature delivery. Mode of delivery, we already mentioned it today. The differences between the C-section and the normal delivery, and the colonization goes through the vaginal delivery route, while the C-section delivered babies need additional time and efforts for restoration of normal microbiota. The maternal vaginal microbiota and overall microbiota, and this is the very important factor. I know more than a few cases when mothers, or mothers-to-be, I should say, pregnant women, were experiencing some infection conditions during their late stages of pregnancy, were prescribed antibiotics, and basically these antibiotics they helped pregnant women to recover, but these antibiotics had really very bad impact on the microbiota. So the children were delivered with dysbiosis. All right, not premature from the examples that I am aware of personally, but with severe dysbiosis. And for these children, the supplementation with bifidobacteria and lactobacilli was suggested by the pediatricians, and that's what they were receiving through the very first days of their life to recover their normal microbiota, gastrointestinal tract. Maternal health status is also important. Obesity, diabetes, inflammation, so all the factors that may imbalance the microbiota of pregnant women, they will have impact on the child. In the obese families, there are high risk of the obese children being born. And this comes, of course, it's a multifactorial thing, and it's difficult to separate whether it is an influence of the microbiota, whether it is an influence of the living habits, diet, or a combination of all of them, and most likely combination. But we do know that the microbiota being transferred from the obese person will definitely have a very strong influence on the development of microbiota in another person towards the obesity. Antibiotic exposure during pregnancy, I already mentioned this thing with a few examples. I personally was aware of, just as a scientist, as a microbiologist, and also working with the pediatricians who were taking care of these cases. Factors affecting the infants, yeah, it's still jumping, not repeating, not circling in the same slide as it happened before. Good to know. Okay, factors affecting the infant's intestinal microbiota during the first 1,000 days of their life. And again, we have gestational age with premature delivered infants. Of course, we have the situations when antibiotics can be used also as a prophylactic treatment to prevent the development of infections. Of course, these will have the severe negative effect on the development of the resident microbiota. Feeding method is also very important. We do know that breastfeeding also is very good for the development of bifidobacteria, and the infants who are from the first days of their life placed on the dietary supplement, artificial dietary supplement, have difficulties with developing bifida. But of course, nowadays we have a variety of supplements enriched with probiotic bacteria. Antibiotic exposure, of course, and we already mentioned that, will have the negative effect on the development of the resident microbiota. And of course, the introduction of the solid foods. All right, the introduction of the foods that may contain in the digestible carbohydrates will definitely affect the development of microbiota, and that will result in the occurrence of the functional gene pools and development of the microorganisms that are similar to the adult microbiota. So whenever the solid food is introduced, it's most definitely point of no return, if you wish, when the transition is going not only feeding the body development, but also in microbiota, converting from the baby to the adult stage. Possible clinical consequences resulted from abnormal colonization. We already mentioned some of the situations, some of the scenarios when abnormal colonization may occur. And these situations are such as the premature birth, C-section versus normal delivery, use of antibiotics in pregnant women, use of antibiotics in prematurely delivered babies. All of these conditions will result or may result in abnormal colonization, either a low diversity, a lack of the essential microorganisms, or even the occurrence of pathogenic forms. Necrotizing enterocolitis often happen with prematurity, asthma, as a result of antibiotic therapy side effects may occur, inflammatory bowel disease, IBS, which may appear with antibiotics, atopic diseases, C-section delivered babies, quite often seen the case. So, probiotics for where do we start? We start with the feeding. We start with the intramammary glands. So, what happens here is that the dendritic cells in the lamina propria, they send dendrites to the gut lumen with tight junction, and they trap bacteria and transport them back to the lamina propria, and from there to the mesenteric lymph nodes, where they can also stay for a few days. And once inside of the dendritic cells or the macrophages, gut microorganisms can spread to some other locations, including mammary glands. And there is circulation from lymphocyte with the mucosal associated lymphoid system. So the first microorganisms that take on message from here, the first microorganisms that the babies are getting are actually the microorganisms which are typical for the gastrointestinal microbiota of the mother. Of course, with the exclusion of many, only those that are transferred can reach the newborn baby. So after I'm here, free from mother, what's happening? What do we see here? What's happening in these scenarios? And we observe here the cross-section of the small intestine of the fetus, and there are two conditions, the fetal condition and neonatal condition, and there are striking differences in the two of them. And you see, of course, I crossed the germ-free fill, which is not true. It's not really germ-free. And it's interesting to mention here because, just a few years ago, maybe more than a few years ago, but still, we were talking about organs and tissues, and even in some textbooks we were reading that in order to function properly, such and such, tissues and organs should be sterile. Now we know that there is no such thing as sterile organs and tissues. Microorganisms are present everywhere. So we have a cross-section of the small intestine of the human fetus in utero versus the newborn. And the fetal intestine appears thin, so it's really underdeveloped yet, and exhibits low epithelial proliferation with a positive gut-associated lymphoid tissues. And when we look at the infant intestine, we have the manifest robust, diverse epithelium, thick layer epithelium with a fast turnover rate and abundant called elements. So definitely the transition, the maturation is there. And why is that important? It is important because this is where the microorganisms will reside. Okay, next slide. So I already mentioned that the dendritic cells interact with microorganisms, and just to zoom into this interaction, especially in a sense of modulation of immunity. And we know several studies show that the commensal microorganisms can evoke T-cell responses through the dendritic cells by binding to TLR2 or TLR4 on the surface of the dendrites. And the commensal microorganisms can also induce the MHC2, CD11, and dendritic cells to secrete interleukin 10, 12, and interferon gamma. And through this way, the naive Th0 cells are primed and they're differentiating to Th1, Th2, Th17, and Treg respectively. So you can see how the microorganisms that are resident to the human body can actually modulate the immune system. We're looking at the probiotics from mother to child, and one of the things that is important to look at here is the relative abundance of the top genera in milk and in feces. And this is where, a lot of information is delivered here, but I wanted, as always, to understand the take-home message from this slide. So, the first important take-home message is that the bacterial transmission is most common during the colostrum phase, and this is when the bacteria with certain amplicon sequence variants, ASVs, enter the newborn baby instant, and they colonize the environment permanently. The The second important take-home message is that the anaerobic microorganisms associated with the gut environment, such as Bacillobacterium, Lautia, some others, they can be and they are transferred from the mother to the infant with the milk. Strep salivarius, we were talking about these microorganisms. Bifidobacterium and lactobacilli are transferred with the milk, and I probably should add here that some of the lactobacilli can be transferred through the vaginal passage, normal delivery, but of course, we should not neglect the most important route of delivery with the breastfeeding. And in combination, the important take-home message is that the most important key microorganisms that are coming from the breast milk, they contribute significantly to the development of the microbiota, healthy microbiota of an infant. Now, what will happen if we will deliver the probiotic to the pregnant woman or to the breastfeeding woman? Can these microorganisms can also be involved in modulation of the normal gut microbiota of a newborn baby? And of course, many scientists pose this question, and we have very good results indicating that the microorganisms delivered to the pregnant women or during the lactation period, and so from the breastfeeding mother to the child, they can also contribute to the modulation, positive modulation of the infant's microbiota. Not to mention, that those who are on artificial dietary supplements, the infants, these formulations nowadays also contain often probiotic bacteria. Some studies, some examples. Okay, for instance, in one study, maternal E. coli numbers correlated with infant's birth weight, suggested the transfer of features to from the mother to the child. Temporary colonization with LGG, and you remember LGG is one of the most studied probiotic bacteria, was possible by giving the probiotic to the pregnant woman before delivery, and the colonization was stable for up to 6 months. I'm not sure why it disappeared. It didn't look as a transient colonization. 6 months is also a very good colonization, especially considering that it's passed from the woman to the child, most likely through the breastfeeding. Another example of the administration of LGG 4 weeks and 3 weeks, 4 weeks before and 3 weeks after the delivery. So the first thing is that the population establishes presence, and then it was continued delivered also with the breastfeeding. It used positive changes and influenced the establishment of the initial bifidobacteria population compared to the placebo. So positive effect of LGG strain on the bifida population in the children through the mother, not directly. It's an indirect delivery of the probiotic bacteria passed through the mother. Another example in the study where the mothers received LGG had a higher prevalence of bifidobacteria and a lower prevalence of adolescents than those in the placebo groups. And it is not clear why. So how LGG may influence the ratios of different bifida in these scenarios. LGG consumption also increased the bifida diversity in infants at 3 weeks and reduced the similarities of bifida microbiota between mother and infant. Now, what does that tell us? Reduced similarities, could it be that the child's microbiota is developing rapidly towards more mature type of microbiota, and how these kind of changes can be driven still remains unanswered. But this is a very interesting result. If we look at the effects of the perinatal administration of probiotics to children, and again we have here, a lot of examples, different studies, and there's always, take-home message, and you have these tables in your handout so you can study them further. But I marked a couple of them for myself. One of them, promises and disappointments. LGG, prenatal 4 weeks, postnatal 6 months. Now, very aged follow-up years, up to 7 years old. And we have significant reduction of the rates of atopic dermatitis. This is like almost at the level of miracle. And then another study, the same LGG, prenatal 4 to 6 weeks, postnatal 6 months. It's about the same scenario, and follow up again 2 years. So these we have good results, but LGG prenatal 4-6, postnatal 6, no differences between the experimental and the control. I don't know why, and when I tried to dig into these studies, the COP study and the Kalimaki study, I couldn't really find any specific, well, there were some differences and similarities, but I couldn't find what specifically was so different that may influence the outcome of these otherwise similar scenarios. With others, we have very interesting results. Not only single strain, but also multi-strain formulations. And again, we have prenatal 4 weeks, postnatal 2 years. Okay, 2 years is quite a long duration. So the eukaryotic host was almost constantly, receiving the probiotic formulation, but still we have significant reduction of the rate of dermatitis in the treated group compared to the control group. Some cases we don't even have the formulations being identified, and this is one of the problems. We still see this problem. These are the 2007, 9, 2001 studies, but even now, papers, manuscripts come to my desk where, some investigators are conducting clinical trials and they're not identifying the microorganism. They're just saying the commercial formulation. And it is very important to know what kind of microorganisms are involved. Another disappointing study with a mixed combination. And, the thing that I want you to notice, when we have Lactobacillus rhamnosus GG, we have these microorganisms identified to the level of the strain. What we have here, like Acidophilus or the next cultures, we don't know what kind of strains we use, and we do know that the microorganisms are different, and different strains of the same species may perform differently. So this is an important thing to keep in mind. Zooming into the things that we were just talking about, all right? Some of these are highlighted to make it more convenient for us to zoom in. So we have LGG 4 weeks before, 3 weeks after delivery. So delivery of the microorganisms through the birth canal, I'm just speculating. Delivery with the milk through the best breastfeeding. We have the change in the transfer and improved development, establishment, presence in the neonates. We have the mixed formulation of different rhamnosus and bifida. And again, the good thing that the strains are indicated here, and we have the increased resistance to the respiratory tract infection after 4 years compared to the placebo group. And definitely the indications were that the immune system was properly modulated. LGG 4 weeks before, 6 months after delivery, reduction of atopic eczema risk, and this is one of these cases. I cannot stop smiling because I think it's almost like a miracle. Up to 7 years, these children were followed for up to 7 years, and there was no high risk, much lower than in the control group, which is absolutely amazing. But at the same time, rhabdosis GG strain 4 to 6 weeks before and 6 months after, no effect. How is that possible? So inconsistency, the take-home messages will be two from this thing. Number one, yes, there are reported cases where we see positive effect. Number two, inconsistency from case to case. In some scenarios it helps, in some scenarios we don't see good results. Can we use this kind of approaches when we're dealing with the medical treatment? The treatment that should go by the book, should deliver the expected result according to the reference book. No, we cannot. So, as much as it is encouraging, it is also to some degree disappointing. And I think the most important thing, and again, I didn't look at these particular cases. I did look at 3 cases from the previous slide, and I'm not a clinical doctor, and I'm not engaged in clinical trials. I was involved in some trials with the animals, lab animals, and animals of agricultural use. So I'm just trying to excuse myself for not being able to understand what were the significant differences, if any, in these trials, which in some cases resulted with absolutely fantastic outcomes, in some cases resulted in no good developments. I don't know. So when we're talking about the probiotics for prophylaxis and treatment, we already saw some hopes and we already saw some disappointments, but I think the important thing we need to look at is the way the probiotic bacteria may work, and in a way this is the answer to the question that was asked regarding the oral action of probiotics versus bacteriocins, and one of my answers was the multiple factors that probiotic bacteria deliver compared to bacteriocins. So I think many more factors will be delivered, positive factors will be delivered with the probiotic as compared to bacteriocin. On one hand, and on the other hand, the concentration of, for instance, the given antimicrobial substance will be much bigger and much more controllable in the case of bacteriocins in formulations as compared to probiotic bacteria. But coming back to this slide, and again, this is a very comprehensive slide, and we see multiple examples. Some of the important things are highlighted here. Undoubtedly, it has been shown that probiotics can prevent NF-kappa activation, and can inhibit the phosphorylation, degradation or translocation of NF-kappa into the nucleus. And whenever the probiotic or other microorganisms are working as inhibiting factors, you can see these kind of signs used here. All right, so this is definitely shown. Other probiotics can increase the NF-kappa activation. So you may say, okay, should they increase or should they decrease? So what is it we want to achieve? And I think the correct answer would be we want to achieve not an I mentioned it before in our first session, we're not talking about boosting the immune system. We're not talking about boosting some things. We're talking about modulation. So, and modulation consists, it's like, you're driving. Sometimes you're accelerating, sometimes you are, pushing the brake pedal, and sometimes you're deaccelerating without even braking, just letting the engine to slow down. And this is the important thing. This is how the microorganisms can and should work, not by introducing drastic changes. And these drastic changes is a key word, especially when we're dealing, for instance, with antibiotics as compared to probiotics or probiotic-derived substances. All right? Instead of delivering drastic changes, we are delivering the changes that work smoothly. Okay? So the important things here, we mentioned the modulation of the NF-kappa B production and the decrease in function in activation. Apoptosis of intestinal epithelial cells is also very important challenge, and it can be prevented, and again, through the modulating action of probiotic microorganisms. And what they can do is to modulate the PI3K AKT pathway. And we have it here, and basically it comes with the changes in phosphorylation levels of P38. And again, we have the activity of Lactobacillus fermentum and VSL3 multimicrobial formulation. The cytokine secretion can be influenced, and of course, at the end of the day, we see the influence on the development of apoptosis. So, altogether, what we have here is the key take-home message. Probiotic bacteria are involved in modulation, signaling pathways in epithelial cells of the intestine. And this modulation goes both ways, by promoting, by activating signals, and by reducing stopping signals. So, and this way, the probiotic bacteria contribute to the normal, healthy function of the signaling systems in the intestinal epithelium. We already mentioned atopic dermatitis as one of the targets which was studied in relation to the action of probiotic bacteria in mothers, in pregnant women, in breastfeeding women, and in the children through the long duration after the child was delivered, up to 7 years. Some of the examples. All right. Here we have other examples, and what do we have? Lactobacillus rhamnosus GG, the strain that we like very much, one of the most studied. We have mixed formulations, and in some of them, the strains are not even mentioned. We have one study where symbiotic formulation is present, and I think it is important to mention here. We didn't talk about prebiotics yet, and I think this is not symbiosis; it's symbiosis. So this must be a type or improper use of the terminology. Prebiotic was probiotic is a symbiotic, not symbiotic. And a good thing again, we have significant reductions in scores, which is the scoring atopic dermatitis in the experimental group. All right, but another example, duration of application for 12 weeks. Okay, some lactobacillus rhamnosus or LGG, and no effect. Rhamnosus, 12 weeks application, significant reduction, 80 weak application. LGG, our beloved strain, no effect. Why? And again, we don't know. So again, the cumulative table that shows various studies in many, if not, not certainly all of them, but in many studies, we have LGG strain used. We have the age of the patient. Of course, the age of the patient varies. And of course, the younger age, the more you would expect good outcome. So here, the first thing that strikes my attention, okay, 50 years old, it's like, old age for, the delivery of the babies. There's no effect, probably because the probiotic was unable to really deliver good action. The immune system of the mother was declining, and the profile of local microbiota in the woman was not really at its best. But then we have 59 patients with 4.1 years, and here we have 5.2 months. So I don't know why it happens really. Is it an age status? Is it the immune system? Really unclear. Effect of probiotics on the incidence of respiratory illnesses in children. And this is a very important take-home message, because we observe the reduction in the symptoms. We also will see the reduction in the absence from the daycare groups. And what we have here is, again, lactobacillus rhamnosus, which is the most studied probiotic. We have bifida BB12, again, most studied probiotic, which you would expect to have really good results. The study populations here, and again, we have the children of the early age up to 5 years old, and in some cases school children up to 13 years old. And the outcomes are really relatively good. We have 27% decrease in the upper respiratory tract infection, 8% fewer cases of symptoms in the treatment group as compared to the control group, which did not receive the probiotic formulation. So all in all, the take-home message on the respiratory illnesses and the ability of probiotics to handle it in children is positive. All children probiotics, some take-home messages: no recommended symptomatic treatment for the common cold in children of the age 6 and younger. So basically, when this kind of treatment is suggested, can be done, but not through the recommendations of the pediatrician. Common cold treatment have potential benefit in older children, that was acknowledged. And probiotics have some promises for the prevention of common cold illnesses in the younger children as well. Clinical trials using probiotics in the treatment of Helicobacter pylori infection, which is a different situation. And again, sadly, what we have here is the conclusion: no convincing evidence on the benefits of the supplementation of probiotic. Temporarily, inhibition of H. pylori disappeared when the administration of these factors interrupted. Very small studies, separate studies, the effect of probiotic treatment in children has been estimated only by the breath test. So what it tells us is that it's the indirect indication, presence of urea in the breath. The H. pylori microorganisms may or may not be present, so it's not the evidence of eradication. It's a symptomatic evidence. All right. So some beneficial effects may occur. They may be strain specific, but otherwise, you cannot really say that there is anything good happening. probiotics with evidence of the positive outcomes, and this is where these effects are separated to the weak, strong, and moderate effects. And what we have here is the NAC, colitis cases, non-alcoholic fatty liver diseases, diarrhea. So many of these cases will be associated with infections, abnormalities in the functions of the immune system and other organs and tissues. And again, our beloved LGGs here, Bifida is there, both in fungus and breathing. We have the multimicrobial formulations, we have yeast, and we have the multimicrobial formulation of VSL3, which is again considered to be quite well studied. Okay, so with LGG and Saccharomyces boulardii, the good outcome is the decline in the duration symptoms, and the strength of the evidence was considered as being strong. With the antibiotics and CDFL-associated diarrhea, again, Saccharomyces boulardii performed really well in reducing the risk of diarrhea with a strong effect. Now, necrotic enterocolitis and LGG, the decline in the prematures but really weak evidence. So take-home message again, inconsistency in the data, and we need more studies which are designed in the same way so they can be compared and the differences can be evaluated. The challenges for the probiotic treatment in the infants with necrotic enterocolitis. Prophylactic treatment in the preterm infants can reduce the instances, but probiotics have not been widely adopted as a standard clinical practice. We have a heterogeneity in the setups of the experimental approaches in the clinical trials data, the durations, times, so really we're comparing apples and oranges. There is not enough data to speak with confidence on the efficacy and the safety of probiotics in populations. And I cannot emphasize enough specific populations because I don't recall seeing, even in the animal studies, and we were talking about it yesterday, what are the factors, what are the genes that start working differently in the immunocompromised eukaryotic host, the genes and the products that may assist the microorganism to go from probiotic to the infection we're dealing with. We don't know, and we're dealing with children, right? Probiotic market is not really well documented, well regulated. I mentioned several times VSL3 formulation. Actually, it turned to be that a lot of companies started to produce VSL3, claiming that they're making the GIAN the only one true VSL3. And when one research group collected VSL3 formulations from different manufacturers, that found that different microorganisms are actually present in these formulations. And then the special message was spread through the community saying that VSL3 is only the formulation produced by the original company and nobody else. All right. So of course we need more standardization and we need more effort to understand better how these microorganisms can help, especially in children. The conclusion remarks, and there are many, all right, and they're coming as a summary of the proposed practice, which comes from the European Pediatric Association. I'm not going through all of them, just the ones that I marked here because I think they're interesting and important. Well, no convincing evidence to recommend the use of probiotics in prevention of gastrointestinal infection in daycare. Probiotics cannot be recommended for the treatment or prevention of atopic dermatitis, regardless of some absolutely fantastic, amazing results with the control over the period of time as long as even 7 years. No safety data on use of probiotics in prevention of AAD in severely ill children. And I think we can't emphasize that enough, the jeopardized immune system and how the microorganisms will perform. And I think, quite frankly, these deserve to be a study which can and should be sponsored by the granting agency, just to model the situation with the immunocompromised, germ-free, for instance, immunocompromised mice. You can get these lines of mice challenged with probiotic bacteria to look at what kind of genes and how they are expressed and what leads to the pathogenic behavior. Some probiotic strains are not recommended to use in children, such as intercoccide, due to the possible transfer of vancomycin-resistant genes. Possible transfer. Are these genes present? Are these genes on the transposon or other elements? So these kind of things must be clarified even before any trials are conducted, and we were talking about it in our previous sessions. Promising interventions for mitigation of diagnosis in the first 1,000 days after birth. Again, we have intervention, prebiotic and probiotic, gut transplant, gut microbiome transplant, microbial ecosystem, therapeutical approach for the infants of gut microbiome target CRISPR therapies. CRISPR, we'll be talking about these methods, very potent new method, but I don't think it will find application in over-the-counter and food products anytime soon, because they are simply not really allowed, and consumers are hesitant to take it, but certainly in medicinal applications. Variety of formulations were developed, and the benefits were shown, such as, in the case of the combination symbiotic use of prebiotic and probiotic, enhanced survival rate of probiotics, improved effectiveness, but of course there are some challenges, evaluation of biocompatibility and safety of hydrogels, which in my opinion are pretty safe, and especially when it comes not to the internal delivery, but topical delivery for sure. And hydrogel formulations can be used, are used in, for instance, vaginal topical applications. Gut microbiome transplants, it is a very important approach, but I don't quite think these can be identified as probiotic, and they're not falling into the category of probiotic. It's just the health promoting action of the healthy microorganisms. And again, we don't have sufficient data on the safety of this kind of approach. Microbial ecosystem therapeutical application, the formulations were designed specifically for the infants who needed to improve their gut microbiota with the support and modulation of the immune system, and of course, again, safety and efficacy in the long-term studies should be done. And with regard to the CRISPR systems, it's engineered probiotics with the targeted applications for the specific purposes. They can and they do help in the model studies, especially the animals, reducing the risk of health issues with some metabolic disorders, and they help to modulate the immune system. But again, some additional studies need to be done to better understand the efficiency of these kind of engineered probiotics in the actual environment of the little creatures. And with that, I would like to finish, and I'm ready to take your questions. Okay.
One in the chat box. Can you see?
Yes, I can see it. Can we expose babies born the C-section way to mother's vaginal fluid shortly after birth? Will it be beneficial for the baby? Can same be done to the premature ones? I don't think that's a interesting question, really. And why to do that? Well, usually C-section is planned in advance, right? Usually it's either by will of the mother or because of the certain health conditions which may require that. And the same is true for the premature delivered. At least sometime in advance, it is known that the baby is going to not to be delivered in the normal way or in the normal expected time. So I think that allows for sufficient time to collect microbiota, and if not to identify and isolate specific health promoting bacteria, but still to use it as a transplant, as the collective microbiota transplant. Whether it will be approved or not, that's a different question. But I think your question actually carries a very interesting idea for the study. And I can clearly see this kind of study, even on the model animals, introduction of the microbiota from the, pregnant, or just giving birth mice to the germ-free. And these kind of studies were conducted, and we'll be talking about it. We'll be talking when we'll be dealing with obesity and the introduction of the microorganisms from which are capable of production of bacteriocins from one organism to another in rodents with the goal to control obesity causing or associated microorganisms to see some good results. I can see these kind of experiments designed with germ-free mice, for instance, also with mice challenged with some non-desired microorganisms, and how the microbiota from the rodents can be transferred. I think it's a very valid question, and I think that these deserve to be studied.
Any other questions? If someone want to ask, can ask directly.
Yeah, you can ask questions. I guess I left the group with more uncertainty than confidence. And to some degree, I think this is a good thing, because really we have more things to understand and to discover. So the lack of the harmony, the lack of the studies that are conducted in the same way. And I think this will continue, because each group wants to study their own things. Not that many groups are interested in standardization across the platform. Is it good or bad? I don't know. The diversity allows for more data to be collected, but lack of standardization is a limiting factor when it comes to making the conclusion.
Yeah. One of them, they are asking how effective is bacterial probiotic against yeast cause bacterial infection.
This is a very good question, because if you will look in the literature, you wouldn't find that many papers that are reporting on the activity of probiotics or bacteriocins against yeast. I did come across several papers, just a few of them, saying that some bacteriocins can also control yeast. If I'm not mistaken, Dr. Todorov had these kind of publications, some other scientists. But the mechanisms are still to be understood. There are different mechanisms. How bacteria work against yeast, I have no answer that will be clear and simple. I think that there are many factors that may be engaged, possibly the very same bacteriocins. Lactic acid, unlikely because yeast can survive really well in the highly acidic environment. If I would be given a chance to work on this kind of study, my first area of curiosity would be form sensing. Is there anything in probiotics that regulate activities in the yeast? That's the first thing I would try to look at.
So if there is no question, I would like to conclude this session, and thanks Dr. Chikindas for really a very informative and clarity in the lectures speech, and the students are, clear in most of the part, but they are thinking out of the box how to ask the question. So that may be delayed. So if they are interacting later, we can do that.
Right, and as Dr. Dwari mentioned, if you will have questions later in time, certainly you're free to ask, and we can come back to the previous topics and discuss them, and to see if we can come up with something that will look as an answer, or at least will shed some light on the problems. Thank you very much, all of you, for your attention, for your time, and I will see you tomorrow.
Have a nice day.
You too. Thank you.
Bye. Okay, anything if you want to ask me regarding registration or regarding course, if any doubt, we can clear it here. Nothing? Okay, thank you very much for joining, and I request further that most of the participants you should join timely. I give only 15 minutes to take the record screenshot. In a 15 minutes, I take the screenshot and send to the IIT Kharagpur National Coordinator. So be careful that you join as soon as possible. The session is going to start. Otherwise, your attendance will remain as such. So we are having tomorrow's sessions in the tutorial at 11 o'clock, where we'll have some demonstration, laboratory demonstration, and also Yakult visit. Tomorrow's tutorial is on the virtual visit to the Yakult industry, which we are having nearby at Sonipat, and also the demonstration of some instruments which are used to work with the probiotics and postbiotics. So with this, we are concluding this session, and thank you very much for joining. See you tomorrow. Good night.